Good morning, students. So today in this lecture video, I will be introducing you to the bacterial growth. My main focus with this particular lecture is your identifying the parameters of growth and also to know the growth kinetics. We need to understand the kinetics of bacterial growth. But before we know the kinetics of the bacterial growth and the depth of it, we should know that what is a bacterial growth. So students, if I differentiate bacterial growth from the growth of other organisms, how will you differentiate that? Bacterial growth is basically the increase in the number of cells. So, bacterial growth is an increasing number of cells, and they basically increase in the number of cells multiplied by your binary fission. Binary fission. So, their protocol for growth or mechanism is your binary fission. Now, you know that this is the typical flow chart of a bacterial growth. Now, you know that in the first 30 minutes, what you get to see is that the cell is actually replicating its chromosome. So, the chromosome is being replicated, followed by the development of a septum that will partition the two chromosomes. That means these two chromosomes will be partitioned at the two sides of the cell, and then the septum will be formed. Now the septum is being completed, and finally you get to see that a single cell divides into two. That is why we are calling it binary, that is 2. Now from that particular individual cell, that is you get to see 2 cells, again the 2 cell is further divided into another 2, so that is your 4 cells. So like this way, at 30 minutes, 60 minutes, 90 minutes, 120 minutes, there is an exponential growth of the cell. So this is how bacteria divides. So from here one can be sure that bacterial growth is no doubt increase in the number of cells. Now we will be focusing on another aspect of your bacterial growth, and that is their growth curve. How does they grow? How does they multiply? So, if you get to see this particular growth curve, it is having 4 phases. One is your lag phase. The second one is your log or exponential phase. The third one is your stationary phase, and the last one is your death phase. So you are not getting to see the x-axis and y-axis. The x-axis of this graph is the time of interval of growth, whereas the y-axis is the log of number of cells. So if I write, just take into account, x-axis is log of number of cells, and y-axis is your time. So next we will move on to the growth cycle. Now if I ask you students that how you will think about bacterial growth, what will you say? You will say that you will take a medium, you will prepare a medium, and in that you will be inoculating microorganisms, adding the microorganisms. But now the next question is that after you add the microorganism, seal your culture tube or your conical flask and put it into the incubator, what will happen? So what happens, that is basically explained by this particular graph. So the lag phase. Immediately after the inoculation, there is no apparent growth. No apparent growth may occur for a period of time. The length of the lag phase is variable and dependent on the period growth history of the cell. It represents a period of adaptation to growth in a new environment and involves the synthesis of enzymes required for growth in this environment. So the cell is basically preparing itself for the cell division. So for that it has to adapt to the environment, it has to prepare all the materials which is required for the cell division to occur, to increase in its number. So all this thing happens in this phase. So that is why there is no increase in number of cell and you have a plateau, you have a straight line. Now the question is that there is a late lag phase where some cell starts to elongate and divide. Okay. So there are many proteins which are also being secreted, like FTSZ protein, which will bring about the septum, mean proteins which will guide the FTSZ to form the septum in the cell. So all these things are basically being produced. Next is the exponential phase. This is frequently spoken as the phase of your balanced growth, where the synthesis of all cells constituent increase at a constant rate so that the cell population doubles and continue to double at regular intervals. This is not a true phase of balance or steady state growth, since nutrients are continuously being consumed and the end product of metabolisms are accumulated. So here you get to see that initially there is a huge volume of medium, huge volume of food. So the organism will multiply exponentially but at a constant rate, right? So there will be a hike in the graph. So you get to see the exponential phase of the graph where the multiplication of the bacterial cell is maximum. Then comes again the plateau form of the graph, that is your stationary phase. This is characterized by no net growth, that is, the death rate becomes equal to growth rate. Please understand this point. You know that when this is actually a culture in which you have not added new medium. So there is a limiting medium. So a time will come when the medium will finish, when the food will finish. So what will happen? The organism will start competing with each other to survive in that particular condition. The cell may remain viable for long periods since the phase with endogenous metabolism, the oxidation of storage polymers, proteins, etc. occurs. The population usually enters the stationary phase as a result of the depletion of some essential nutrient and the build-up of toxic product. So, you know, the organism will compete and produce certain secondary metabolite as a weapon to kill other organisms. So there is start of the death of organism, but here the growth rate is equal to death rate. So there is no net increase in cell number. As a result, you get a plateau in your graph. Then comes your declining phase. Declining phase means it started to decline. So during the stationary phase, the death rate may become higher than growth rate. So when your death rate is becoming higher than growth rate, then there is a declining phase. And ultimately, there is occasionally referred to as deceleration phase. The deceleration phase is important since growth is unbalanced and the growth rate varies as a function of residual substrate concentration. So whatever the residual substrate is there on which some organisms are surviving and most of the organisms are dying. And as a result, there is a drop in the graph. That means there is a deceleration phase or decelerated graph of the whole. So thus, from this particular graph, we get to see it is the lag phase, the log phase, the stationary phase and the death phase. And death phase goes down till the number becomes nearly nil. Again, you may be asking one question, that why the phase of the growth has not started, the graph has not started from the zero. This is because when you add certain inoculum into the culture and you start the whole process, there are certain number of cells in the inoculum, in the culture, in the medium. And as a result, we cannot say that the graph should start from zero, that is no cell at all. So this is all about your growth curve. And depending upon the growth curve, the nature of bacterial growth, now we will move on to the kinetics of your batch culture. So, next we will move on to the kinetics of the batch culture. Now, before we move on, we first need to understand that what is a batch culture. Till date, what we are discussing in the growth curve is a batch culture. Why? Because the system is closed. There is no exchange of energy or mass with the system. You have taken one conical flux, you have added a medium, microorganism, stoppered it. That's it. So the organism is growing over there and it is utilizing the medium and finishing off the medium. So this is about a closed system of bacterial growth, bacterial culture. So that is basically a batch culture. So first point is unicellular microbial growth is autocatalytic, that we know very well, such that the rate of growth is proportional to the cell concentration already present. So there has to be some initial number of cells. That is why I am focusing on that point, that the graph doesn't start from 0. We are adding some cell and that is the initial concentration of the cell. And the growth will be proportional to the initial concentration of the cell. So, if you see this particular equation, you get to see x0. What is x0? It is the initial number of cells. Then it doubles, as I have talked about in the first slide, that is 2x0. Then it is 8x0. So, this is your nx0. N is the number of times, number of cells. So, n is that particular multiplication factor, 2, 4, 8, like that. So, nx0. So, like this way, it will go on, continue its division, and it will have the n will term as the number of generation. So, now know, x is the cell dry weight that is given in gram per liter, and the interval is constant, termed as doubling time. Why I am calling it as a doubling time? This is because in each particular cell, each particular time, you know, that one cell doubles. One cell doubles into 2. So that is why the time is called as doubling time, right? We can also call it generation time. That is the time taken by the organism to move from one generation to another generation. So, if you get to see this equation, n is equal to T by Td. N is doubling, number of doublings, n, after time t. T is the total time, and what is your Td? Td is an individual doubling time. So, it follows that the cell density, xt, what is xt? xt is the cell density. Xt is the cell density after the completion of the growth at time t is related to x0. What is x0? x0 is the initial number of cells we have started with. So, we can write in this way, xt is equal to x0 into 2 to the power n. Why? Because xt is the final cell number. So, how this final cell number comes? It is the initial cell number into the number of generation. Number of generation the organism has multiplied to come to xt. So, that is why we can also write xt is equal to x0 2 to the power n. Now, what is n? N is t by td. So, we can substitute n by t by td. So, 2 to the power t by td. Now, you rearrange using logarithm. So, you take log at both sides. So, here it will be log of xt by x0, just cross multiply, is equal to log of 2 to the power n, that is 2 to the power t by td. So, now if you simplify it, ln xt minus ln x0, take t by td at the front, t by td log 2. Now, you take the t, again you cross multiply, you get to see ln xt minus ln x0 by t is equal to ln 2 by td. Now, what is ln 2? ln 2 you have to convert it into log 2. So, it is 2.303 into log 2, that is log 2 is 0.3010. So, if you multiply, you get to see it is 0.693 by td, right? This is more commonly expressed in this term, that is 2.303 log of xt minus log of x0 by t is equal to, you have already converted ln2 over here, that is 0.693 by td. So, in conclusion, we can say a plot of log x versus time yields a straight line with a specific slope. And what is that slope? That slope is 0.693 by td. So have you understood from this particular explanation? It's simple because here you get to see that there is two important parameters, that is your xt and your x0. xt is the final cell number, x0 is the initial cell number, and you know that 2 is that particular multiplication factor by which it doubles, right? It's a doubling time. So xt will of course be 2 into x0 into 2 to the power n, where n is equal to t by td. Am I clear? And then you just simplify the equation. Now, if you convert it into differential form, you know, it is d by dt of ln x. That is equivalent to 0.693 by td, which we have already got. However, if you further differentiate, d by dt ln x is equal to d ln x by dx into dx by dt. So, and since d by dt ln x is equal to then 1 by x dx by dt. So, we can write 1 by x into dx by dt is equal to 0.693 by td, which we have got. So, that is equivalent to mu. So, what is mu? Mu is the specific growth rate. We can simplify the equation as d by dx is equal to mu x. So, equation is termed the growth equation, where mu is the specific growth rate defined as the rate of increase in cell concentration per hour. Equation in this differential form, what is the significance? Form of the growth equation and can be used to describe growth at any stage during the growth cycle. So, it can explain any stage of the growth cycle by this particular equation. In special case where xt by x0 is equal to 2, that means when it has doubled, right? The cell population doubled. So, the equation becomes mu 2.303 log 2. Just put x by xt as 2. So, it becomes log 2. So, ln 2 means 2.303 log 2 by td, right? And that is equivalent to 0.693 by td. So, we get to see the ultimate equation, that is mu is equal to 0.693 by td. So, this is about your batch culture kinetics. Now, I will move on to the continuous culture kinetics. Now, continuous culture kinetics, before we move on, we first have to understand that what is continuous culture. How can you differentiate with the batch culture? Continuous culture is a continuous system where both the mass and the energy is always being transferred. So, in continuous culture, there is an inlet and there is an outlet. Through inlet, you insert the medium and the inoculum, and through outlet, you remove the waste product. So, what is the advantage of continuous culture? The advantage of continuous culture is that you will always have no deficit of medium. You will always have enough medium for the microorganisms. So, microorganism will move into the exponential phase and it will be constant at the exponential phase. It will not move to the stationary phase. But there are many other parameters to be taken into account while we are talking of continuous culture. The first important thing is the flow rate. Flow rate of the medium. So, flow rate. So, rate at which the medium supply enters into the vessel. That is your flow rate of the medium. So, as the flow rate increases into the vessel, what will happen? There will be increasing washout of the cell from the vessel. So what will happen? Because of the increased washout, washing out doesn't mean just the substrate washout. The cell also washouts. So as a result, the concentration of the cell decreases in the vessel. So that is your dilution rate. So we have to control the flow rate and the dilution rate to maintain the constant number of the cells, such that our fermentation is completed within the stipulated period of time and with that accuracy, and it should be completed and it should be optimum. So that is why we need to regulate the flow rate and the dilution rate. So that is why we first have to understand the kinetics behind it. And this is basically the first point which I will be discussing over here, is the relationship between the dilution rate and the cell concentration. So I am talking about again and again about the dilution rate. So that will bring about the change in the cell concentration, and if there is a change in cell concentration, right, it may affect your fermentation. So that kinetics need to be understood. So first is the rate of flow of medium into a continuous culture system is shown as dilution rate. So it is called as your dilution rate. So now if you see the equation very carefully, we are trying to find out the cell accumulation in the vessel. So for that, what we are taking into account? Cell in. That means how much cell is moving inside the vessel. Next is your cell out, minus cell out. That means how much cell is going out of the vessel. Then plus growth. That means some cells are growing inside the vessel. So that has to be added. So plus growth minus death. So those cells which are dying in the vessel. So if you do this equation, if you frame this equation, ultimately you come to the conclusion of your cell accumulation. So if I move into the derivation, dx by dt, that is rate of change of cell concentration per time. Because here I have to take dx by dt, because in every interval of time the cell concentration is changing, because it is an open system. Okay, so dx by dt is equal to fx0 by v. What is f? F is the flow rate of medium supply. What is x0? X0 is the initial number of cell in the medium supply. So the seed tank which is actually giving the cell into the fermentation vessel, that is your x0 by V. V is the volume, constant working volume of the bioreactor. Next is your cell out. So from where the cell is going out? Cell is going out from the bioreactor. So Fx by V. So F is the flow rate of the medium supply. X is the amount of cell inside the bioreactor. See, this x0 is being entering into the bioreactor and x is inside the bioreactor. So Fx by V. Next come mu x. That is, mu is the specific growth rate. How much growth is going on, right? So of course the growth is, we are focusing the growth on the vessel itself, the organism inside the vessel itself. So it will be mu x minus alpha x. Alpha is the specific death rate, and x of course is the number of cell in the vessel. So now you can understand, or you can relate this particular individual terms with respect to the equation. Now you have to simplify this equation. So if you think, it can be simplified by the thing that is mu is very greater than x. Of course it is a continuous culture. Growth rate will be very greater than death rate. Why? Because otherwise the cell will not be in exponential phase. We know that it is in exponential phase because there is no deficiency of the medium. So mu is greater than x. Consequently, the equation can be simplified. You just neglect the death rate, this one, and Fx0 by V, because now what we are trying to see is the X accumulation of the cell inside the vessel. So now you just remove this two term or neglect this two term, you get dx by dt is equal to minus Fx by V plus mu x, that is the reduced equation. Now what is F by V? Flow rate per unit volume will ultimately indicate to dilution rate. as I have told in the first part of the derivation, that if you don't control your flow rate, ultimately the dilution rate will increase and that will bring about your washout. So of course flow rate per unit volume is of course your dilution rate. So you just put F by V by D, just to substitute F by V with D. So dx by dt is equal to minus D plus mu x. So you take common x mu minus D. So this is during the non-steady state. So when every time the cell concentration is changing. But what is during the steady state? When the rate of change of cell concentration is 0. It's constant. So when dx by dt is equal to 0, what do? You just substitute 0 and you see that it will be mu is equal to D. That is, your growth rate becomes equal to your dilution rate. So the equation shows that simply by varying the medium substrate rate, medium supply rate, that is your flow rate, the growth rate can be varied. The growth rate changes. This will be correct until dilution rate is greater than that of your maximum growth rate. Okay? This will result in the fall of cell concentration and washout of the culture. So you understand that if your dilution rate becomes more than your maximum growth rate, what will happen? It will result in the excessive washout of the cell from the vessel. So that is what they are saying, right? So then from here we can determine DC, that is your critical dilution rate. The critical dilution rate is defined as the lowest dilution. So we have to take into account the lowest dilution rate at which the washout will just start to occur. So we need to understand that when the washout starts, so that is your critical dilution rate. And in practice, your critical dilution rate is your maximum growth rate. So if there is a maximum growth rate, there is a huge concentration of cells in the vessel. So what will happen? If there is a flow rate, increase in the flow rate, since there is so many cells, some cells will actually come out as along with the metabolites, along with the excretory metabolites from the other end of the fermenter. So the washout will occur. So that is why they are saying that the critical dilution rate, in lowest dilution, if you get to see the washout, that means what? That means there is a maximum growth rate in the vessel. So critical dilution rate is equal to your maximum growth rate. So this is about the relationship between dilution rate and cell concentration. So in my next class, I will be again discussing the relationship between the dilution rate and another aspect which is also very important in the continuous culture, that is your substrate concentration. So in the next class we will be discussing dilution rate and substrate concentration. So if you have any questions, any queries about the derivations, about the kinetics, please write in Google Classroom and we will be having a discussion. Thank you once again.